Our next speaker is Clarissa Weaver, and the title of her presentation is Untangling Neurodegenerative Disease.
What's the last thing you got really tangled? It happens to me all the time with my earbuds, but it could also be fishing line or thread in the sewing machine or maybe something like Christmas lights. It's frustrating, right? In all cases, you can't use the thing that you wanted, the thing that's tangled, and you know it's going to take time and energy to sort out the mess. It's not just a problem for us, it's a problem for any living cell. See, inside of every cell there are proteins, and these proteins have to have a certain shape or structure to perform their necessary function. But if the cell gets damaged or stressed, these proteins get misshapen, and then they can get tangled together. We call these tangles aggregates, and they're deadly to the cell. That's true if it's a bacterial cell, a plant cell, or even a human brain cell. In fact, protein tangles are implicated in a number of neurodegenerative diseases, including Parkinson's disease, Alzheimer's disease, and ALS. There are some treatments available currently, but they mostly just treat the symptoms. You're not getting at the underlying disease. So if we're going to stop or reverse neurodegenerative disease, we need an approach that will clear out or reactivate the proteins trapped in those toxic tangles. I study a molecular motor, a protein called HSP104, that rescues proteins from tangles. It clears out the mess in the cell, and it allows those proteins to return to their normal shape so they can perform their necessary function. Now, the motor that I study comes from yeast, but the same motor is present in bacteria and in plants, but not in animals, not in humans, which leads to the question, could we use this untangling motor to stop or reverse neurodegenerative disease? And in fact, recent results have shown that with just one slight modification, this motor was able to protect neurons from damage in a worm model of neurodegenerative disease, specifically Parkinson's disease. So this motor actually prevented neurodegeneration. Now, the focus of my work is on understanding how. If we can understand how at the molecular level this motor is able to break up protein tangles, then we can tune this motor for therapeutic application. Prior to our work, most researchers in the field thought that because this motor is shaped like a ring, it must pull that damaged protein through its center. You can picture pulling an entire thread through the eye of a needle little by little. And there are other similarly shaped motors that do work that way. But if you picture trying to pull a tangled thread through the eye of that needle, you might suspect it wouldn't work as well. And in fact, what I found in my work is that that's not how this motor works. This motor takes just 2 or 3 steps, tugging and pulling, likely on the loose parts of that tangle, causing the knot to fall apart, which is probably more similar to how you'd untangle that ball of Christmas lights or your earbuds. Now, if I then asked you, how could we make this untangling machine better? You'd probably say, make it faster. But what I found is that for effective untangling, slower is actually better. When you have this motor with modifications that make it more effective at combating models of neurodegeneration, it actually has slower activity. So imagine taking that finding and applying it to a specific target, such as alpha-synuclein, which forms tangles in Parkinson's disease. So the next time you have tangled Christmas lights or earbuds on your hands, I hope you'll think of my work with the molecular motor HSP104 and remember to slow down. Thank you.